So here it is another kind of quorum sensing which is shown in Vibrio Harveii. Vibrio fishery is a very familiar name because Vibrio fishery generates the illuminations, and in this Vibrio Harveii is also another strain of bacteria which generates this quorum sensing, and quorum sensing is really hugely studied in this Vibrio Harveii. They do not generate any kind of luminescence, but still they are very effective. Why they are really important? Because to study Vibrio fishery, we can say a very important facts about quorum sensing, which is not known to us by only looking at the normal gram-negative and gram-positive bacteria having the quorum sensing mechanism. In the Vibrio fishery, this is an overview of how Vibrio Harveii interacts with its own different cells, and also as well as they can interact with the other cells of other species or the other strains of bacteria. In this case, what we are looking at here, this is the plasma membrane here. This is the inside of the Vibrio fishery, Vibrio Harveii. Inside of Vibrio Harveii, and this is the periplasmic space. So don't think it is external space. This is the periplasmic space, and after the periplasmic space, we have the extracellular space or external space, like that. Okay. So in this picture, what we are looking at here, there are different names out there. Don't puzzle by the names. This is a very simple pathway as we are looking at. Normally, it has also the technique of the quorum sensing. The basic idea remains the same, that they have to have a huge density of cells to produce the autoinducer molecules, which is designated here AI1 or autoinducer type 1. Normally, what we have discussed earlier is all regarding with this autoinducer type 1. So what this autoinducer molecule, which is type 1 molecule, normally does, it generally binds with this receptor, and when they bind with the receptor, and the receptor has some structure, the structural conformation shifts, and finally it triggers to produce, it activates another set of proteins, that activates another set of proteins, and finally the gene, which is a structural gene or the functional gene, which will produce the effector molecule or the protein, the effector protein, or the proteins which effect can be easily seen outside. This is called the effector protein or effector gene. So finally, with the actual goal of all this thing to transcribe this effector gene to produce the effector molecules, which sometimes help to make luminescence, sometimes helps to induce virulence factors, sometimes helps them to colonize, to make biofilms. So in this case, this is the pathway which is known to us, that the autoinducer molecule which is produced by these cells can bind with this structure, with this receptor, which have a several transmembrane region like that. In this picture it is shown clearly. After the attachment, there are different change occurs in the conformation. Finally, this is called the Lux N protein. So change in Lux N, and then the change in Lux U protein. So normally Lux U protein is get phosphorylated. So the protein gets phosphorylated. After the phosphorylating event, it phosphorylates another set of protein, which is Lux O, and finally this Lux O protein bind with this DNA, and finally transcribe the DNA, translation is done, and the protein or the effector protein is made. So this is a process we all gone through. Okay, so in this whole system of mechanism, we have to have a kinase domain. So in this case, what we have the kinase. Normally tyrosine kinase is present there as a receptor of this autoinducer molecule. So throughout this process, it is a phosphorylation event goes on. When the phosphorylation event goes on, when there are lots of amount of this autoinducer molecule present there. When there are not such amount of autoinducer molecule present, the whole system will be blocked, and the reverse system is going on. To turn on this event reversely, we need to dephosphorylating all these parts. That's why we need to have different sets of enzymes, not the kinase. We need to have phosphatases. Normally, this enzymatic subunit of the proteins are designed in such a way that they can also function as kinases, that they can also function as phosphatases. This mechanism can be better understood in this direction. So let me delete it is all this. Yeah. Now let's talk about this pathway where we have the autoinducer molecule 2. So normally it is noticed that autoinducer molecule 1 is generated to have a signal between the own strains or between their own genera of bacteria. So suppose there is Vibrio Harveii. So Vibrio Harveii wants to talk with another Vibrio Harveii, wants to talk with another member of their same family. They produce the autoinducer 1. So this autoinducer 1 molecule is highly specific for a particular strain. Okay, but it is noticed in other case the autoinducer 2 type of molecule, which is structurally different than autoinducer 1, and also it functions differently than autoinducer 1, is there to have a signaling between different strains of bacteria. For example, Vibrio Harveii wants to talk with Salmonella or wants to talk with E. coli, then it will trigger the second pathway or the pathway of autoinducer 2 throughout this place. Okay, so in case of autoinducer 2 production, the same process is going on. The autoinducer 2 is produced. There are lots of other type of bacterial cell outside. So lots of autoinducer 2 is made, and this autoinducer 2 want to attach with the receptors here. In the autoinducer 1 pathway, what we have seen is that the molecule directly attaches with the receptor. But in this case, it usually directly not attaches with the receptor. It attaches with the receptor via a mediator here, or carrier molecule here. In this case, the carrier molecule is the Lux P. It's a protein which is a carrier protein of autoinducer molecule 2. Autoinducer molecule 2 binds with Lux P protein, and the Lux P protein finally attaches to the receptor side, and finally it will trigger some change in the kinase region, and then the whole system is going on. But in this case, the system is considered in slightly different manner than the previous scheme. So in this case, here again the autoinducer 2 molecule attaches with Lux P, and the Lux P is then attaches with the receptor, and finally it triggers the kinase activity. Lux Q is activated, Lux Q phosphorylates Lux U, and Lux U phosphorylates Lux O, and finally Lux O produces the effector molecule via transcribing the Lux CDABE. These are different genes, CDAB, different sets of genes are transcribed to make different proteins, not one protein but different sets of proteins, which helps them to maintain the virulence in some case, or in some case to colonize them to make biofilms and all these things. Okay, and in the reverse order, they can go via the phosphatase activity. When the phosphates are cleaved, they can grow throughout the reverse order, and finally the system will release this autoinducer 2 molecule. Okay, so this is the basic concept about the two types of pathway. So it is noticed that the autoinducer 2 pathway is very, very important in very different cases, because a bacteria not only needs to communicate with its own family members, but it also needs to communicate with other members of other family members to transfer the virulence factor, to transfer some resistance genes which they have each other. So that's why they need to develop this system. So this system can be found. So let's now study this autoinducer 2 system and about this Lux P protein. So what this Lux P protein actually do? It is a binding protein, as we know. It binds with the autoinducer 2. Where it binds with autoinducer 2? And more importantly, what is the autoinducer 2 molecules? So what types of molecules are called the autoinducer 2 molecules? What are the structure of autoinducer 2 molecule? And all these things are derived via different techniques, different processes. Normally scientists isolate these protein molecules, and finally they crystallize them to finally get the structure of the protein molecules. As this is not so easy as I am telling you. It's really hard work, but still after doing all these things, finally they crack the structure of this autoinducer 2 molecule, and as well as the molecule of Lux P, because the Lux P attaches with the autoinducer 2 molecule to make a complex, and this complex is the thing, this complex is the only able thing to attach with this receptor. If there is only Lux P, it will not bind to it. If there are only autoinducer 2 molecule, that also fails to attach with this receptor. Okay, so they need to have, and all these different proteins, which are the receptor proteins, always the integral protein, and they are transmembrane, because they need to have region which will span the membrane inside and outside both to have a proper signal. So now let's talk about the next here. So for that. So what makes this autoinducer 2 molecules? What are the different types of proteins that are made in autoinducer 2 molecule? You can see here it is made by the Lux S gene. Lux S gene. And this concept is this kind of research work is done a few years ago, just 1 or 2 years ago, and scientists tell us that there are Lux S genes out there which will code for some proteins. Those proteins are able to make the autoinducer molecules. And what are the chemical components of autoinducer molecule? Okay, and these are the different types of bacteria that those are having the Lux S system, Lux S gene. And so those molecules which are having Lux S, those bacteria which are having Lux S gene, definitely can go through the autoinducer 2 system. So now let's look into the system, how this whole thing occurs. Normally there are there are different processes of several genes. So there are several genes, for example, it's a gene concept. So if we look at this gene system, we can find different regions in the gene. For example, this is a gene, and this gene we have different regions. We have for example, this is the mate gene, and then we have this PFS gene, and we have methyl transferase gene, and we have all this gene, methyl transferase gene, PFS, and methionine activating gene or degrading gene, and all these genes are there and to utilize this whole pathway. Okay, so what happens, where we are now, let's discuss. So it's a bacterial cell. This cell will produce some autoinducer molecules. Okay, so to make this autoinducer molecule, we need a set of genes. Normally in the previous pictures we call it the Lux I genes, remember, in this Lux I genes, or now we are calling it Lux S genes. So whatever we are calling it Lux I or Lux S gene, this gene, this set of genes, there are not one gene, there are different sets of genes out there. The sets of genes are producing some molecules which will make the autoinducer molecule. Not only the autoinducer molecule, but it also have to make this molecule, also have to make this Lux P molecule, this kind of molecule, because it needs to have a carrier molecule which will bind to the autoinducer 2. So in this case, when it makes all those molecules and finally it will transcribe and translate, the molecules are made, then only the whole system of autoinducer 2 system can be carried on. In this picture, if we are looking at this, we have meat and we have made kinase which will phosphorylate that to make SAM. Okay, so so here we have the methylated adenine. Okay, so methylated adenine portion is there. So methylated adenine portion, we need to transfer this M from this place to another, or methylation region or methyl groups from this molecule to another molecule, different molecules, to carry on the signaling. So we need to have methyl transferase enzyme which can be coded via another gene here. Okay. After that, the SAM is converted to SAH, or the homoserine residue system, like that. Okay. So adenine with the homoserine residue in this case. Okay, so this adenine with homoserine residue, this total molecular sequence is toxic to cells. Try to understand this process very well. So what we have the receptor portion, and it is kinase is made, and finally it will transfer the methyl group from the SAM into another molecules, and finally it will results to produce the SAH, or what it is called. It is the adenine with the homoserine residue, and this adenine with the not homoserine, this homocysteine, not homoserine. So adenine with a homocysteine residue. So adenine with homocysteine residue, but this is toxic. Why? Because this adenine with homoserine residue molecule can block the previous step via the feedback inhibition. It will block via feedback inhibition machinery. So we need to convert this molecule into some other types of molecule which is not so toxic. So the same thing is done by the product of PFS gene. Is the PFS gene, the product of PFS gene convert this adenine homocysteine residues into the ribose homocysteine residue. So the adenine is converted into the ribose homocysteine. So ribose homocysteine residue means there is a ribose sugar and there are the homocysteine residues out there. So cysteine, there are lots of cysteine residues out there. That's why I call the homocysteine. Okay. So this is a control. We can look at holding down this system. So what we are doing in this case, we are making some products. That products are toxic. It acts like toxic. It can block the whole system via the feedback inhibition. So we need to convert those toxic material into some non-toxic material, and we are also doing this via a product made by the same type, this set of genes, which is PFS in this case, to finally makes a non-toxic molecule which can be placed, which can be stable. But what is shown in different experimental setups, we never find this SRH molecule in the system. So in whole this system, when the system is performed, when the whole reaction is performed in vitro situations, we never find this SRH molecule. That triggers a question in our mind: where this SRH molecule is gone? Instead of this SRH molecule, all the time we end up with the formation of homocysteine and 4,5-dihydroxy-2,3 pentane-dione molecules. This is called DPD, or dihydroxypentane dione, and along with that we form the homocysteine molecules. So never SRH molecules. So this is a basic concept, that somehow this SRH molecule is converted into homocysteine and DPD molecules. So what is the material which helps to convert this homocysteine, this SRH molecule into homocysteine and DPD molecule? Okay. And what are this DPD molecule and homocysteine molecule are doing in this solution, or doing in the normal event of cellular machineries? There are many, this molecule, this DPD molecule is observed many years ago, but nobody knows the function of this DPD molecule. So what scientists have done few years ago, they just take these DPD molecules and add them in vitro situations. They give all the necessary equipments, bacterial cell and all these things, some bacterial cells there, but very few amounts of bacterial cells. What they see, that this DPD molecule, due to the presence of higher concentration of DPD molecule, the higher concentration of DPD molecules in vitro situations, for example, let me erase all this, let me erase all this ink. And now this DPD molecule, you have this DPD molecule, so scientists just extract this DPD molecule from the environment. You have a test tube, we add this DPD molecule, in presence of a few amount of bacterial cell, and as a result of it, they produces luminescence in case of Vibrio fishery. So what we are looking at in this case, though they have a very few amount of bacterial cell, but still they are able to produce that luminescence. How they produce this luminescence? How can they produce this luminescence due to the less amount of bacterial cell? The only way of producing this luminescent effect due to the presence of higher concentration of autoinducer. So the only reason in this case that helps this bacterial cell to produce the luminescent is DPD. So this kind of experiment suggests us that this DPD molecule is nothing but autoinducers. So we call it the autoinducer molecules. So what kind of autoinducer molecules are they? Normally, due to via different sets of experiment, we came to know these are the type 2 autoinducer molecule, which can be found. So this autoinducer molecule along with homocysteine can be found. Okay, but most of the time, why are not we are now after doing that, we need to have a structure of this homocysteine molecule along with this DPD molecule. Okay, so to do that, we need to extract those homocysteine molecule and DPD molecule from this tract and need to look for the structure. So in this case, we use this and finally look for their structures, and we'll discover their structure. But before going to that, one question still remains there, is what is the product which helps to convert this SRH molecule into the DPD and homocysteine residue? And the product in all these cases, there are another sets of experiment is carried out. What are the experiment? We have a test tube, we add the SRH molecule in the test tube, and we look for the conversion of SRH SRH into homocysteine and DPD molecule. and after some time we extract all these things, we remove homocysteine and DPD molecule, finally end up with some products, some molecules. Those some molecules can be produced, definitely is produced by the bacteria, but what kind of gene produces these molecules? To know the genes responsible for making molecules that converts the SRH into the DPD and homocysteine, we need to go through another approach. The approach is the classical genetics approach, or the classical genetics approach. The approach is via the mutation, creating mutant. So in this case also we are making mutations. We are making mutations in different variable genes of bacterial cells. So we take a bacteria, for example Vibrio fischeri or Vibrio. Normally this whole thing is performed in case of the Vibrio Harveii, and in this Vibrio Harveii, what the scientists have done, they take some genes. So here we have again, let me draw the gene sequence. So here we have MED gene, for example the MED gene. Here we have the transferase gene. Here we have PFS gene, and we also have another sets of genes in the same lineage, in the same lining. For example, we don't know what the gene is and what products it makes, but we know the previous genes and what products they made. So what the scientists have done, they mutate this gene. They make a mutation. They make a point mutation in a single amino acid sequence, change in a single amino acid sequence, and as a result of it, this the division of SRH into homocysteine and DPD is blocked. And then they suddenly identify the gene, this gene which causes some protein. That protein helps the SRH to be converted into homocysteine and DPD. So then they name this gene they name this gene Lux S. So they name it LUSX. Okay. So this is the gene which is designated by scientists that may have the effect on converting SRH to homocysteine and DPD. So now it's the time for testing this hypothesis. So what they have done, they take this Lux X gene and they transcribe this gene. They make a Lux X protein. So you have a Lux Lux S. So made a Lux S protein. This protein is added into a test tube. So in a test tube we add SRH, then add some Lux S protein, and after some time of incubation, what we have seen these products, homocysteine and dihydroxypentadiene or DPD. Okay, so this is a very moment. This is a moment of clap. Something is discovered, is really interesting, really fascinating. Something which is not known for many years is finally discovered after some time, that this part of the gene is nothing but. So let me erase all this. This part of the gene is nothing but Lux S. So this is the protein which helps to convert the SRH into homocysteine and DPD molecule. Okay. So after cracking the code of the protein, finally now we have to discover the structure of this Lux S as well as the DPD structure. So to go into that, we need to go through different sets of portions, and to discover the structure, the scientist encounters another major problem. What is the problem? Normally what they are looking at when the whole system is going on in vitro, these homocysteine and DPDs are made, but after the making of this homocysteine and DPD molecules, what happens is SRH, which is the ribose homocysteine residue. This is fairly unstable, not actually unstable, fairly very active. This molecule is really very active molecule. Very active molecule. This is very active molecule that they can be converted into some other molecules. So all the time the ribose ring, in this case what we are looking at, so this is the pentameric ring of ribose sugar, as we all know. This ring is attached to this molecule, and this ring is really unstable and very reactive, that all the time the rings is broken, and this breaking of the ring and the joining of the ring. So the ring and the linear form can be formed throughout the time, and the mechanism is so that the equilibrium is so dynamic, the equilibrium is so fast, that there's a problem for scientists to actually understand this whole structure of this SRH when it presents in this situation. After many years of research, active research, finally the scientists from different regions, from different parts of universities, the crystallographer scientists crack the code of crack the structure of this molecule. They finally able to crystallize this molecule, and finally after the crystallization of the molecule you end up with the structure something different. So this is the structure of this SRH molecule, or the molecule. Here is the ribose ring and is the homocysteine residues out there. Okay, so in this case this is converted by Lux S into DPD and homocysteine. So these are the. So normally the DPD is present in this form. In many times the DPD can be formed in other forms. So there is an equilibrium. So this equilibrium is so rapid that it's very difficult to snap the structure of a protein via different techniques that are available of the molecular structurization. Okay. So after many years of research, after many times of research, we finally developed, finally crystallized the structure of this molecule. And finally, when we crystallize the structure of the molecule, we make something like that. Okay. So here is the Lux P. Remember what is Lux P? So let's go backward down our slide. Here is the Lux P. So Lux P is the carrier molecule. Remember the carrier molecule. Because we need a carrier molecule which will bind to the autoinducer 2 to carry the autoinducer 2 from the outer space throughout the periplasmic space, and finally attach it with the receptor. So Lux P molecule presents in the periplasmic space of those bacteria. And what is the structure of the Lux P? So normally after many years we have a chance to actually crystallize the structure of Lux P along with that autoinducer molecule which is presented in the middle here. So this is the autoinducer molecule you can see in the middle. So it's like a pocket in the middle. So there is a small or tiny pocket in the middle of this Lux P protein. So Lux P protein is made of a different several alpha helices. Because it's a transmembrane, it's a periplasmic protein. Always it has to be present in the membrane. That's why it is made up with different alpha helices. And in the core of this molecule, there are loop-like structure. In the core of this loop structure, we have this autoinducer molecule. So this is the autoinducer molecule 2, because as we know, in case of only the autoinducer 2 molecule, you need to have the carrier molecule, which is Lux P in this case. Okay, so now let's zoom on the structure. If we zoom on to the structure, finally in here, this is the different residues, different cysteine residues of Lux P, which will interact with this autoinducer molecule. What is this autoinducer molecule? If we look at the autoinducer, which is the DPD, in this case, this is made up with the different rings. 2 rings are there, 2 pentameric rings are there, and these rings, the red colored molecules are the oxygens, and these are the methyl groups out there. This black colored molecule are the methyl groups out there. Okay, and here is another moiety over there. What is this moiety? For the first time, normally people think about it as a carbon, but if we think about it as a carbon, the whole system cannot be done, or the bonding cannot be explained. After some time, we finally came to know that this part of the region is not made up with carbon. This is in fact another molecule which is very abundant in the marine water, is boron. Okay, so the presence of boron again thumbs up for our hypothesis, for the hypothesis of scientists, that this kind of system, this kind of auto-inducing factors is present in the marine water, because Vibrio fischeri and Vibrio Harveii, both types of bacteria are found vigorously in the marine water. And in the marine water there are lots of boron atoms out there, and those boron is engaged to make this ring. And finally you have boron, and boron have a tendency to bind with the hydroxyl residue. So there are different hydroxyl residues, it will bind with the hydrogen bonding throughout this place to maintain the structure of this autoinducer molecule in the pocket made by the Lux P protein. So this is electron density structure of this Lux P hollow enzyme, and in the middle here we have the autoinducer molecule, which is much clearly viewed in this case. So here it is the formation strategy of autoinducer molecules. So what we have this DPD molecule here. So DPD is in turn is not act as an autoinducer molecule. It is further folded, because as I have told before, that this DPD molecule is very reactive. it will fold, it will just open its structure and fold and open and all this time throughout this place. So it folds to make a DHMF structure like that, and finally it folds, it refolds again with the presence of water. The hydrolysis is done and the bonds are made and break, and finally the borate salt is there, and the presence of this borate salt, the boron atom adds and to make a final ring, to make the second pentameric ring, and it will attach to the hydroxyl residues here, hydroxyl molecules there. The one thing I recommend you to notice in this picture is that the orientation of hydroxyl and methyl groups. In all this case, the hydroxyl group present in the upward and methyl is in the horizontal line. So this is the presence. So the hydroxyl here, methyl here. Okay. So now if we look for different sets of molecules, not AI2, AI1 pathway. If we look at the AI1 pathway, we can find the orientation will be different. The orientation will be methyl and hydrogen and hydroxyl like that. Okay. When we will discuss the picture, we will discuss it again. Okay, so these are the folding steps are going through. The first thing, it will need water to make the cyclization. After making the cyclization, the dehydration takes place, and as well as the dehydration, it will have to have the borate salt. Finally, to attach the boron into the top here, the top position here, and finally makes the THMF borate molecule, which acts as the autoinducer 2. So this is one type of autoinducer 2 that is designed. There are lots of different varieties of autoinducer 2 molecules can be found here, but in most of the cases in the marine waters we find boron in this place as autoinducer, make autoinducer.